In this video, we are going to talk about some traits that don't follow the Mendelian patterns of inheritance. We call these non-Mendelian traits. Before we do that, I wanted to mention something that I meant to include in the previous video. That is the idea that the dominant allele isn't necessarily the most common allele in a population. You could have a trait where 99 out of 100 people are homozygous recessive, and one person out of 100 has a mutation that makes them heterozygous, which means that they have the dominant trait. If that individual mates with one of the homozygous recessive individuals, about 50% of their offspring would be expected to have that dominant trait. But when any of the other 99 homozygous recessive people breed with each other, none of their kids are expected to have the dominant trait. Overall, a very small proportion of the offspring of the entire population would be expected to have the dominant trait. One trait that works like that in humans is polydactyly. Polydactyly is when a person has extra fingers or toes. There are a number of different genes that are involved in whether or not a person will be polydactyl, but at least one of the mutations that causes polydactyly is dominant. That means that a person only needs to have one copy of the allele of that gene that causes polydactyly in order to have extra fingers or toes. So even though polydactyly can be a dominant trait, only one baby out of every 400 or so has polydactyly in the US. This is a gentleman from Cuba who has 12 fingers and 12 toes. Polydactyly is also common in cats. I honestly don't know if polydactyly is a dominant trait in cats, but I couldn't resist the urge to show you a cute cat picture. The traits that we've talked about up until now, including polydactyly, follow the rules that Mendel discovered for inheritance when he was studying his pea plants. Since the time of Gregor Mendel, we have learned that while many genes follow the rules that he discovered, many genes don't. We call genes that don't follow the rules of Gregor Mendel non-Mendelian traits. Some non-Mendelian traits don't show that relationship of a dominant allele versus a recessive allele. In many cases, perhaps the majority of cases, a gene has more than just two alleles out there. It is also possible and very common for a mutation in a single gene to change the phenotype in multiple different ways. Sometimes one gene will affect the expression of another gene, and often a trait is controlled by multiple genes, not just by one gene. In other words, genetics is actually a lot more complex than what was discovered by Mendel, even though the discoveries that he made were true for a certain number of traits. Mendelian traits, or traits that follow Mendel's rules, exhibit complete dominance. This means that if the dominant allele is present, the offspring will have the dominant phenotype. You won't be able to tell the difference based on the phenotype between a homozygous dominant individual and a heterozygous individual. A homozygous dominant purple flowering pea plant is going to look the same as a heterozygous purple flowering pea plant. They're both going to be purple. In incomplete dominance, neither of the traits is able to completely suppress the other trait. If you took two parents that were homozygous for two different alleles for this trait, the offspring would have a phenotype that was somewhere in between the phenotypes of the two parents. This flower is a snapdragon. There is a gene that helps control flower color in the snapdragon that comes in two different versions. One of the alleles will make the flower color red, and one of the alleles will make the flower color white. Notice that we have used a little bit of a different notation system here. We're not using a capital letter and a lowercase letter for the two different alleles because we don't have one that's dominant and one that is recessive. These alleles exhibit incomplete dominance. We have used a capital letter C for color to represent this gene, and then we've used a superscript to indicate what color the allele is for. Capital C with a superscript capital R means the color that this allele codes for is red. Capital C with a superscript capital W means that the color that this allele codes for is white. If this trait showed complete dominance, you would expect to see the hybrid offspring of these two homozygous individuals to show the dominant trait. So if red was the dominant trait, then the offspring would be red. If white was the dominant trait, then the offspring would be white. In incomplete dominance, however, a mixture of the two traits is present in the heterozygous offspring. We call this an intermediate phenotype. In the case of the snapdragon flower color, an individual that has one allele for the red version of the gene and one allele for the white version of the gene will be pink. When that individual from the F1 generation goes to reproduce, half of its gametes will have the red allele for the gene, and half of its gametes will have the white allele for the gene. If you cross one F1 hybrid individual with another F1 hybrid individual, you will get the Punnett square on the bottom. Remember that in peas, with the completely dominant purple flower color, you would have seen 3 of these individuals be purple and 1 of them be white. Here you can see that the heterozygotes have a different phenotype from either of the homozygotes. So you have one red individual that is homozygous for the red allele. You have 2 pink individuals that are heterozygous for the red and white alleles. And you have 1 individual that is white that is homozygous for the white allele. Another way that a trait can differ from the Mendelian laws of inheritance is if it is codominant. In codominance, the 2 different alleles for a gene will both show up if an individual is heterozygous for that gene. In incomplete dominance, what we saw was phenotype mixing, so red and white became pink. In codominance, what we see is both phenotypes being present at the same time. For example, there is a gene in cattle that controls hair color. An individual that is homozygous for the white allele of this gene will be white. An individual that is homozygous for the red allele of this gene will be red. And an individual that is heterozygous, meaning it has one white allele and one red allele, will be what we call roan. Roan means that it makes both white hairs and red hairs. The cow isn't pink, so it's not incomplete dominance. The cow makes both red hairs and white hairs, so the phenotypes of both the red and white alleles are showing up. Another way that a trait can differ from the Mendelian laws of inheritance is if it has more than 2 alleles for the gene. One gene that fits into this category is the gene that controls what your ABO blood type is. The A allele of this gene codes for the addition of a certain carbohydrate to the outside of your red blood cell. We'll call it the A carbohydrate. The B allele of this gene codes for the addition of a B carbohydrate to the outside of your red blood cell. The little I version of this gene codes for the addition of no carbohydrate to the outside of your red blood cell. In addition to having more than two alleles, this gene violates the Mendelian laws of inheritance by exhibiting a certain degree of codominance. If an individual has both the A and B alleles for this gene, they will make both A and B carbohydrates on the outside of their red blood cells. The A and B alleles of the gene are codominant to each other. However, the little I allele of the gene is recessive to both the A and B alleles. The phenotype of an individual that has two A alleles will look the same as the phenotype of an individual that has one A and one little I allele. The same is true for an individual that has two B alleles. Its phenotype will look the same as an individual that has one B and one little I allele. So the relationship between the A allele and the little I allele, or the B allele and the little I allele, is one of complete dominance. Thus, you can get a mixture of codominance and complete dominance even within the alleles of a single trait. Up until now, we've mostly talked about genes that have one effect in the body. In reality, a lot of genes are involved in multiple different pathways and processes, and so altering one gene can lead to multiple effects in the body. When a gene has multiple effects, we call this pleiotropy. One gene that can have pleiotropic effects is the beta globin gene. Beta globin is a component of hemoglobin, which carries oxygen in your blood. A mutation in the beta globin gene can lead to a condition called sickle cell anemia. In sickle cell anemia, the beta globin mutation leads hemoglobin molecules to stick together within the red blood cell. The long rods formed by hemoglobin molecules that are stuck together can bend the red blood cell out of shape. They can start to form a crescent moon shape. There is a farming tool with a curved blade that is used to cut grain that is referred to as a sickle. So that crescent moon shape, or that sickle shape, is what gives the disease sickle cell anemia its name. This one mutation in the beta globin gene can lead to multiple effects in the body. It can contribute to blindness, it can contribute to liver failure, and it can lead to a heart attack. So we say that the beta globin gene mutation has pleiotropic effects. In pleiotropy, we said that one gene affects multiple traits. It's also possible for multiple genes to affect one trait. We call this polygenic inheritance. A commonly used example of polygenic inheritance is skin color in people. Skin color is determined in part by how much melanin you have in your skin. Melanin is a pigment that is able to protect the skin from UV damage from the sun. Generally speaking, people who lived near the equator of the Earth, meaning that they were exposed to the highest levels of UV radiation from the sun, evolved the ability to produce more melanin to protect their skin from those damaging rays. People who lived farther from the equator and were exposed to less sunlight did not develop as much melanin because they needed the sunlight to help them produce a vitamin called vitamin D. The amount of melanin that an individual has is determined by multiple different genes. As a result, we get this beautiful spectrum of human skin color that doesn't fit neatly into a set number of categories. Sometimes, when a trait is controlled by more than one gene, one of those genes will affect the expression of another of those genes. We refer to that as epistasis. One example of epistasis is the interaction between two genes that control hair color in Labrador retrievers. One of these genes, the one that is shown here as big B or little b, controls whether the pigment that is produced by the cells in the Labrador retriever's skin will be black or brown. Again, this pigment is a form of melanin. The other gene controls whether or not the pigment that is produced by the skin cells of the Labrador retriever will be deposited into the hair of the Labrador retriever. In this image, the capital letter E stands for the dominant trait of depositing the pigment into the hair. The lowercase letter e represents the recessive trait of not depositing the pigment into the hair. If this trait followed Mendel's laws of inheritance, you would expect all of the offspring who have a big B allele of the pigment color gene to be black, and any individual that was homozygous recessive or had two copies of the little b allele of the pigment color gene to be brown. However, if you look at the four squares in the bottom right, you will see that three of those dogs have a big B allele, so you would expect them to be black, and one of those dogs has two little b alleles, so you would expect it to be brown. What you have instead is four dogs that are yellow. That is because the gene that determines whether or not pigment will be deposited in the hair is affecting the expression of the gene that controls pigment color. The four offspring possibilities in the bottom right side of the table are all homozygous recessive for the gene that determines whether or not pigment will be deposited in the hair. As a result, it doesn't matter what kind of pigment the dog is able to make, meaning black or brown, that pigment won't end up in the hair. There's actually another gene that controls the production of another type of melanin that can be either yellowish or reddish. So the genetics of hair color in Labrador retrievers is actually a little bit more complicated than we're showing here. Finally, an organism's phenotype may not match the phenotype predicted by Mendel's laws of inheritance because environment can also play a role in what phenotype is expressed. To go back to the idea of skin color, the skin color that is expressed by any given person on any individual part of their body will be different depending on whether that part of the body has been exposed to sunlight in the environment. So traits like skin color aren't just a product of the genotype of the individual, they're also a product of the individual's environment. Another example of the ways that environment can influence the expression of the genotype of an organism is when individuals don't get enough nutrients, and that results in them not growing to their full biological potential. That often happens in human children, but here you can see it happening in a strawberry plant. The strawberry plant on the right doesn't have enough phosphorus. As a result, it can't make enough things like phosphate groups that are in ATP, for example, and it isn't able to grow. The strawberry on the left could be an identical genetic clone to the strawberry on the right, but since it has access to phosphorus, it can grow to be a lot bigger. So the genotype can sometimes determine the potential for what the organism could be if the organism has access to all of the nutrients and resources that it needs. We can use the genotype of an individual to predict the phenotype that they will have, but we can't know for sure what that phenotype will be because that organism will grow up in an environment that may or may not have the full complement of resources.